Hello everybody, my name is Feiner and I am a PhD student at the Saarland University. Our research is on aging and DNA methylation. Today I will present to you some interesting results of our recent work. The title of my presentation is HOXA5 Methylation Changes Correlate with Age and Gene Expression. As most of you know, the aging process has been associated with differential patterns of DNA methylation, which also relate to changes in gene expression. Therefore, we aim to identify genes that are differently methylated in old and young individuals. We approached our aim by performing genome-wide methylation analysis, followed by mRNA and protein expression profiling of the genes we aim to find. After all, our goal was to find a correlation between the DNA methylation and gene expression of a specific gene of interest. We started our approach by collecting blood samples of young and old volunteers and isolating monocytes from these samples. Next, we isolated genomic DNA, mRNA, and protein from the monocytes. After a bisulfite treatment of the DNA to convert unmethylated cytosins to uracils, we performed a genome-wide methylation microarray. The microarray we applied analyzes the methylation status of over 850,000 known CpG sites. These are predominantly CpG sites located in tumor-relevant regulatory regions such as promoters and CpG islands. Based on this approach, we identified CpG sites which were significantly differently methylated in old and young volunteers. We assigned the associated genes and selected interesting candidate genes based on criteria such as a methylation difference greater than 20%, a p-value under 5%, and the genomic location of the CpG site. For example, promoter, gene body, and 3' UTR. Subsequently, we performed a local deep bisulfite sequencing of the selected CpG site, also called BPOF. This step serves as a validation because here in the microarray we only have about 10 to 20 reads per CpG site, and here in the bisulfite sequencing we have over 1000 reads per CpG site. Finally, we investigated the gene expression of the candidate genes on the mRNA and protein level by real-time PCR, ELISA, and Western blot analysis, respectively. This slide depicts a selection of genome-wide results of the microarray. Although overall methylation is known to decrease with age, the methylation at the regulatory CpG sites, as analyzed by the microarray, tends to increase with age, as you can see on the left picture. The right picture illustrates the annotation of differently methylated CpGs, short DMCG, in young compared to old individuals. In this picture, the left pie chart shows the distribution of the DMCG generated by the genome-wide methylation array. As you can see in the two right pie charts, the DMCG, which are located in CpG islands, are more often hypermethylated than hypomethylated in old individuals. And we have more DMCGs in non-island regions that are hypomethylated than hypermethylated in old individuals. After performing the genome-wide methylation analysis, we assigned the CpGs to the associated genes. We chose 3 interesting candidate genes with a methylation difference greater than 20% and a significant p-value. As illustrated here, the genome-wide methylation analysis indicated for all 3 DMCGs significant hypermethylation associated with old age. The 3 genes comprised dual specificity protein phosphatase 3, short DUSP3. This protein negatively regulates members of the MAP kinase superfamily, which are associated with cellular proliferation and differentiation. And we have homeobox5, short HOXA5, anti-angiogenic member of the HOX family, and ryanodin receptor 1, short RUR1, a protein found primarily in skeletal muscle. We also selected them as candidate genes because the CpG sites are each located in different gene regions. The target CpG site of DUSP3 is located in the 3' UTR. The CpG site of HOXA5 is located in the promoter in a CpG island, and the CpG site of RUR1 is located in the gene body. After validation with the local deep bisulfite sequencing, only the target CpG of HOXA5 showed a significant methylation difference, as you can see here. Next, we performed real-time PCR on the 3 genes. It turned out that the methylation result was reflected in the mRNA expression analysis in the way that only HOXA5 mRNA expression is significantly downregulated in old individuals. This result is in line with the age-related hypermethylation in the promoter region of HOXA5. Next, we investigated the protein expression of HOXA5. As illustrated here and here, the HOXA5 protein expression is also significantly downregulated in old individuals, which is well in line with our HOXA5 mRNA expression results. As it is known that HOXA5 interacts with p53 and may upregulate the expression of p53, we also examined the expression of p53 in our samples. Since p53 protein is not expressed in monocytes, we only measured p53 mRNA expression. As shown here, p53 is also significantly downregulated in old volunteers. In addition, we see a middle strong to strong correlation between HOXA5 and p53 mRNA expression. p53 is also known to interact with p10, an effective suppressor of cancer and a contributor to longevity. Hence, we investigated the age-dependent p10 mRNA expression. In line with the p53 downregulation, we observed a reduced p10 mRNA expression in old age. We also investigated correlation of p10 mRNA expression with the mRNA expression of HOXA5 and p53. As you can see, we observed a middle strong to strong correlation. Finally, we studied the protein expression of p10, which was also significantly downregulated in old individuals and correlates with HOXA5 protein expression. In summary, our results are as follows. First, we have shown that methylation at regulatory CpGs tends to increase with age, although total methylation decreases with age. Second, our methylation analysis indicated an interesting candidate gene, HOXA5, whose methylation at the target CpG site is significantly increased with age, while its gene expression is significantly reduced. And this reduction takes place on 2 levels, the mRNA and protein level. Moreover, we were able to show that the age-related regulation of HOXA5 gene expression correlates with gene expression of downstream targets p53 and p10. Our findings have shown again how important it is to validate the results of a genome-wide methylation analysis by local deep bisulfite sequencing, as we observed that after the validation, the number of putative candidate genes has become less. Our main result, that age-related upregulation of HOXA5 methylation correlates with its reduced gene expression, qualifies HOXA5 as an eligible aging marker. We further conclude that HOXA5 hypermethylation in advanced age contributes to p53 downregulation, and in that context may also contribute to the increasing age-dependent cancer risk in the elderly. I am now at the end of the presentation, and thank you very much for listening.